Hi, everybody. Welcome again. It's a great pleasure to have with us Dr. Mette Olufsen from North Carolina State University. Professor Olufsen did her PhD from Roskilde University in Denmark in 1998, modeling the arterial system, cardiovascular stuff, working with Charlie Peskin during that time, and then did a postdoc in Boston University, working with Nancy Koppel on neuroscience, and then went back to cardiovascular stuff that she's been doing for quite some time now. So we are looking forward to this talk on the use of computational models to understand cardiovascular system dynamics. Over to you.
Thank you very much, and thank you guys for inviting me. It's always great to get an opportunity to talk about my work. And please feel to ask any questions or put it in the chat as long during the talk. I'll be happy to stop and answer the questions. So the first thing I was just going to make sure of is that you guys all know how the cardiovascular system works. So basically we have a heart, and I'm going to turn around on my Zoom. And I'm going to raise up my hand. I don't know if you know it, but this is my right hand and this is my left hand. So what you can see here is that the left heart is always depicted on the right side. The right heart is depicted on the left side. So we have blood at high pressure being ejected from the left heart. It is feeding all the organs in the body. And then there is an exchange of oxygen and carbon dioxide. And then deoxygenated blood is transported through the systemic veins at a low pressure back into the right heart. And then in the right heart, there's another pump acting, and that's putting out deoxygenated blood into the pulmonary arteries at about 20 millimeters of mercury. Again, we have an exchange of oxygen and carbon dioxide, and then it's returned in the pulmonary veins. So the heart really acts as a pump, and it's a pump that's driven by changes in pressure. So studying this system from a computational perspective, there is a lot of different questions that you can ask, and I've just listed here some of the questions that my group has been working on. Some of them we are going to be addressing today, and some of them we're not going to put so much emphasis on today. So one question you can ask is, could we do in silico, and that means using the computer, treatment of high blood pressure? This can be high blood pressure in the systemic circuit, high blood pressure in the pulmonary circuit, and what happens in the response to various treatments. Another question that we have been working on that recently got a new grant to study is, what is the effect of the aortic remodeling? And the aorta is this main vessel that comes out of the left heart. Is in different types of babies that are born with only one functioning ventricle. So these are called the Fontan babies, because instead of having a two-sided pump, you rewire the system to a one-sided pump. And another question you could ask is, if you have a lot of blood clots in the pulmonary circuit, which is the one up here, and you want to use microballoons to insert stents, and assume you have 20 clots but can only insert 3 stents, where should you insert those stents? And an opposite question that we recently started working on is, a lot of small dogs, including my own dog that's now lying in the bed, are born with shunts, especially the small designer breed dogs, that bypasses the liver. And it's not great, especially if you're a dog, to not have flow going through the liver. It's not good if you're a human either. And so then some of the questions we are asking, how should you place that stent? That's not a stent, that's a constrictor, because you want the flow to not go through bypassing the liver. Where should you place that shunt? How should you place that shunt? What happens if it's not functioning the way that it should be functioning? And then the final type of question I'm going to address here is a slightly different question. So the first type of question we study using a fluid dynamics type of model that predicts a periodic flow and pressure waveform. But we all know that blood flow and pressure varies over time. And so sometimes they do a lot of functional tests of the cardiovascular system, which is, how does the system actually respond to changes? And so, for example, you could be interested in what patients can tolerate certain types of anesthesia and which ones cannot. How do you differentiate patients that have issues with fainting. So a big group of those patients are those with long-term COVID. They have a lot of issues with controlling their blood pressure, and we are trying to gain more insight into studying that. Okay, so if we start with the first type of models, which are really based on studying fluid dynamics in some form or shape. So the fluids dynamics could be solved in like 3D, CFD, or FSI models. So CFD meaning we are just using a tool calculating the fluids in 3D, but the wall is not moving. And if we say FSI, we mean fluid structure interaction, which means you're solving the 3D equations in a deformable tube. We are not, my group has not put a lot of emphasis into that. There are a lot of groups out there that are really good at that. So we are going to focus more on the COD models. The COD models really thinks about the blood flow as an analog to an electrical circuit. And that started really with efforts put out by Guyton in the early 70s. He created these elaborate models of the cardiovascular system. Or you could think of it as a 1D network model. We have illustrated one here down below, where we are solving the fluid dynamics equation, but we are going to assume that every vessel can be represented by a tube, and we are only looking at that solution in the radial direction. The tube has a radius, and we can get that from data. So that's the focus on this particular talk. And one of the things that my team really, no matter the problem we are looking at, is how do we incorporate models with data? And there is, in this type of analysis, a lot of different types of data that you could think of. But for all the models based on fluid dynamics, we really have two types of data. We have data that comes from images, and then we have data that comes from hemodynamics. And typically in clinical measurements, image data could be like CT scans or MRI scans, and hemodynamics data could be 4D MRI, which is this funny terms that the clinicians love, but it's really just you have a velocity field and you look at it over time. Or it could be measurements of hemodynamics with a pressure catheter, with a FenaPress device, with pulse wave EKG. So there's a bunch of, or even just a pressure cuff put on your arm. So those are the hemodynamic data. So if we start with the CT scans or MRI scans, which is often referred to as magnetic resonance angiograms because it's a still image. It's to differentiate them from the 4D MRI. You get images that looks like that. Using image segmentation, and that's really an art in itself. And I say art because when it comes to segmenting networks, it's really, really challenging. And the current AI methods are really not very good at it, especially if it's a lung. So here's a rendered image of the arteries, which are blue, and the veins, which are red, of a human lung. Now, once you have a rendered volume, if you want to solve things in 3D, you need to mesh it. If you want to solve it in 1D, you need to extract a network or a graph, which we do by placing center lines in the graph and in the 3D rendered volume, constructing a graph. From that, we construct a connectivity matrix and labels representing the radius of each vessel. I'll get to more discussion of that later on. And then we do fluid dynamics modeling, and then we could predict your fusion or wave intensity or whatever is a question that's associated with the application. So starting with getting the center line, here we are really talking about a mass that really has to do with geometry. There's two prominent approaches used for that. The first approach often use some kind of version of what is called the vascular modeling toolkit. There's a website for that. It places spheres in the vessels and it finds the center of the sphere, and it makes those sphere match through the vessels, and then it predicts the center line as the radius of the sphere. One of the challenges with this approach is that often it puts the junctions where the junctions shouldn't be. And this particular software, if you have complex networks, often will create branches where no branches are, or it will have not be able to capture all the vessels in the network. So we have spent more effort using what is called skeletonization. So skeletonization is a methodology where you take this rendered volume and then you keep removing voxels, which is pixels in 3D, from the side until you only have the center line voxel left. That has a challenge that sometimes you will get a vessel, if there's a small inaccuracy on the 3D rendering, it will think there's a vessel here and generate a vessel here. So we need to clean this graph that's constructed after skeletonizing the image. So here are some of the challenges with skeletonization that luckily you can use graph theory to really remove. So one problem is that sometimes we have branches where they shouldn't be. So for example here, that we go in and look at the image, the 3D rendering, look at the skeleton, and we can see where it is. It's typically in the big vessels. Another thing we almost often see is the root of the vessel is not a perfect cylinder. And so therefore this automated skeletonization algorithm will generate this Y here. And we just wanted to start right here at this point right there. Another issue is that these 3D rendered images, or really the MRI scan, has a finite resolution. And so sometimes the rendered image will assume that this vessel is connected to this vessel and create a circle where there is no circle. In fact, in the vascular system, there's almost no loops. And so what you can do is a graph is represented by all the edges or all the nodes having a degree, which indicates how many vessels goes in and out of it. And it's very easy once you have constructed the graph to spot these circles, and you can use different versions of Dijkstra's algorithm to remove those circles, and then you get your network that we can use to form a connectivity matrix. So the next step is to identify the radius of each vessel. So if we are assuming that the vessel is in the lungs have relatively short vessels and they are mostly straight. So how do you find the radius for the vessel? Well, from the graph, we have a radius estimate at each point. That's obtained by finding the shortest distance from the point in the skeleton to the background. However, when you are in this region that represents the junction, the concept of radius is not really well defined because you don't have a radius in the junction region. So what we did was we used statistical change points to identify changes in this radius estimate we have at each voxel along the vessel. And then we found the change point. So the red dots here denotes the change point. So it's an algorithm that you just let run on each vessel. You have to limit the number of change points per vessel. We set it to maximum 3. And then we found the part of the vessel that had between change points that had the smallest slope. And then we took the mean of that and the standard deviation of that, and we assigned a radius and a standard deviation for each point along the entire vessel. And what was kind of nice is when we looked at these radius estimates, we could create a probability density function, and we noticed that for all vessels, it was mostly normal. So that was the data that allows us to generate a network. Now comes the hemodynamic data. So an example of the hemodynamic data is the 4D MRI. So 4D MRI is a velocity field that's generated after they do the image sequencing where they get the radius. And if you superimpose the angiogram and the 4D MRI, they're not on top of each other. We are interested in volumetric flow, and the way the volumetric flow is generated is they integrate the 4D MRI over the velocity field over the 4D MRI. But if we are not paying attention to the fact that the MRA and the MRI are not on top of each other, you get very wrong estimates of volumetric flow. And it's because you're breathing and you're moving a little bit in the scanner. So with Dan Lear at Baylor College of Medicine, we developed a registration algorithm that basically moves the 4D MRI onto our MRA, and then we were able to extract volumetric flow that you can see. Here's an example of volumetric flows at each of these slices along the vessel. Now, an important point when you're doing modeling is that our model assumes that flow is conserved. And if you're not bleeding, flow will be conserved, which means that the flow going in has to equal the flow going out. But if you take the data, that doesn't happen. And that's because a lot of these 4D MRI data, they have really a lot of uncertainty associated with it. So we set up a linear system that says the flow in has to equal the flow out. And then we added these Lagrange multipliers alpha. We could estimate them. And then we scaled the flow so that we used the minimum possible scaling. And that allows us to align the data so that we had conservation of flow. Now, is that really good to do to modify the data? I know our statistics friends are not very happy about that. But if you don't do that, it's very challenging to really get the models to actually act and fit well with the data. So now when it comes to the fluid dynamics equations, we are solving a 1D model derived from the Navier-Stokes equation. It assumes conservation of mass, which basically says the change in area over time has to equal to the flow in minus the flow out. We have a momentum balance, which is really force equals mass times acceleration. And then we have that has three dependent variables, which is area, flow, and pressure. So we need another equation, which in this case is a linear elastic constitutive law. And to assume that vessels get a little bit stiffer or act stiffer, then they get smaller, we have this relation here that Young's modulus. So when the radius gets smaller, this quantity gets stiffer. So I tried to highlight the model parameters in different colors. So the green ones, which are the radii, we get from the imaging data. The rho and the delta, which is a boundary layer thickness and a density, they are just set. We are not estimating them, but the vessel stiffness, we are trying to see if we can estimate given the model and the data. And so that's the next couple of things that we're going to talk about. So since this is a hyperbolic system of differential equations, we have boundary conditions, we have junction conditions, which I didn't talk about, and then we have boundary conditions at the end of the network. So if we just go back here a second, you can see here we have a network. So when one vessel splits to two, we have conservation of flow and continuity of pressure. And then when we get to the very terminal, we need to add a boundary condition. So what we are doing here, we are assuming that at the end of each vessel, they really continue branching until you reach the capillary levels. And the way we are assuming that these branches some kind of a fractal manner. So we are assuming that the parent vessel branches into two daughter vessels that are scaled relative to the parent vessels with alpha and beta. In the small vessels, we're assuming that viscosity dominates the flow, so we can get rid of the nonlinear term in the momentum balance equation, and we can get a linearized equation that we can solve analytically or semi-analytically. And so that provides us with an impedance that is used at the boundary of our network. So now here comes an example or a couple of example applications. So the first application is we are comparing predictions of flow, not only in the vessels where the data was measured, but also in the organs, because that's what they really worry about with these patients. So these are kids, they're about 10 to 12 years old. They have a single functioning ventricle, and we have 4D MRI. We have some pressure measurements from a cuff. And then we have these images from them. And we are interested in understanding what happens in two different groups of kids with a single ventricle. So the one group are born with both the aorta and the main pulmonary artery emanating for the right heart. So if we go back to the very first slide we have here, so instead of having the aorta coming out of the left heart, the aorta and the main pulmonary artery comes out of the right heart. They have done some reconstruction in these kids, making this guy not go into the heart, but being attached to the systemic veins, but they don't have to do some remodeling of the aorta. The other subgroup of these single ventricle patients are born with their aorta attached to the left heart, but the left heart is underdeveloped, so it can't pump any blood. So those kids, they basically do plumbing when the kids are born, moving the aorta from the left to the right heart. That's the functioning ventricle. Those two types of kids both live into early adulthood. Eventually, they will all need a heart transplant, but we want to postpone when they get a heart transplant as late as possible. But this group that had the aorta moved don't do nearly as well as the group that doesn't have the aorta moved. So what we are studying is what is really happening with the growth and remodeling in the aorta of this group versus that group. So here you can see the radius of the aortic arch is very different in this group than in this group. And what does that do to the flow and pressure? And could we possibly go in and do another surgery, remodeling, including maybe a synthetic aorta when they're 10 or so, and will that help? So that's the question. We set up our model. It has a lot of parameters. So now here what you want to understand, we have an inflow, we have outflows, we have split our domain into 4 different groups. Group 1 is the aorta, group 2 is the arms, group 3 is the left side, and group 4 is the right side. And then we were trying to estimate these model parameters in 4 patients of each type. And then we are going to try and see if we can see differences between the 2 groups. And so first, the model have 40 parameters, but we only have data in 4 vessels. And we clearly cannot estimate all of these parameters because it's too many of them. So our first step was to conduct sensitivity analysis. And what is sensitivity analysis? So we want to match the prediction of our model to the data. So we generate a least squares residual. That's the one we call R here. So that's the model minus the data at the locations where we have data. So 4 flows and 1 pressure. And then we want to see how that residual changes when we change the model parameters. And assuming that we are log scaling the parameters because they have different orders of magnitude, then the derivative of log is 1 over theta. So that local sensitivity is the RD theta times theta. We could also do sampling and do more of a global sensitivity. The problem with the local sensitivity is that they're evaluated at our current parameter values, which we don't know are right or wrong. So maybe instead of just evaluating at the current parameter values, you can sample and evaluating it over the complete on the range of the parameter values. So one way of doing this is using Moise screening. So it samples. It takes a lot longer to compute. You can also use Sobel indices. That takes too long to compute. So we did the Moise screening. So basically you get individual effects and mixed effects. So here's an example of the result of our sensitivity analysis, and that's showing in the log scale. And unfortunately, most of our parameters have some sensitivity. There are a few here that are less sensitive. These are listed by group and averaged over the 4 subjects. And so we then said, okay, all the parameters are kind of sensitive, but some of them are correlated. So for example, when we have this function here for the stiffness, if we only have 1 measurement, we can't estimate both K1, K2, and K3, so these parameters are not identifiable. So we did covariance analysis, so we created a correlation matrix from our sensitivity matrix. We used singular value composition followed by QR factorization, and those of 2 both gave us a tentative subset. Again, this is done locally, so it's a little bit challenging to see if this is still a subset over the whole parameter space. But it gave us an idea for what parameters that we could estimate. We tried to estimate them. We ran what is called a multi-start parameter estimation to check that if we change the nominal parameters that we estimate a little bit, do they reach the same estimated parameters? And that you can see here. We were lucky.
Excuse me. Can you go back to the slide before?
Yes.
Like these parameters, K3, KS3, etc., you have numbers on the x-axis. Are these the different branch numbers?
No, these are groups. So remember,
Groups?
we generated 4 different groups of our parameters here. So we made all the parameters in vessels in each group the same.
Okay, I see.
But
Thanks.
you really have a parameter for each vessel, but that's impossible to do. So we limited our parameter values to groups. And so we said all the groups of this, all the vessels of the same type should be in the same group, which means they have the same parameters.
Okay, got it. Thanks.
Okay. And so the 1, 2, 3, 4 are the groups. We have all the K3, KS3, that's the stiffness in our boundary condition, and K3 is the stiffness in the large vessels. So we ended up deciding that we could estimate all the K3 parameters, both in the large vessels and the small vessels, and the K2 in the small vessels, but not the large vessels. And alpha and beta, that's how this fractal tree changes. So clearly, if you change both of them, that's not an option. You can only change one of them because they're very sensitive, but they are also not both identifiable. But the nice thing was that we started our optimize. These are all the parameters. They're all scaled from 0 to 1. So they're easy to see. We started it at random initial values, and then we ran our parameter estimation. And you can see for all these parameters, we were able to receive identifiable results. And the optimizer here, we used a built-in optimizer in MATLAB, and that worked. We have tried things with different optimizers. The optimizer also matters which one you're choosing, and how you scale the parameters matter. I think here we scaled each parameter with its nominal value, so they all started on being on a scale from 0 to 1. Okay, so here's an example of our predictions for one of the DORV patients and one of the HLHS patient. So the black dots with arrow bars on is the model. The red line is the prediction, and the gray bar is it says the flow data. The gray lines are the flow data, and this shadow gray lines is the spread we had before we started estimating to make sure that our model lied with it, that the parameter range we are sampling from will be within the model solution. And we have recently, and I'm not going to talk about, developed a physics-based neural network model that was also able to do this. And notice here for the pressures, here we had only measurements of the systolic and diastolic values. We did not have any measurements of the waveform. So we were very happy with the results. And so now we could compare some of the predictions from the model and some of the parameters. And one of the things we observed, which kind of makes sense, that there is much more variance in the parameters from the HLHS patients. And remember, we had only 4 patients in this study. And in general, they had stiffer vessels, which is what we expected. But also there was much more difference between the patients. So it was hard to do statistics. We recently got funded to redo this with about a data set with about 200 patients. So we just started working on that. We saw that the pressures are definitely different. The systolic pressure is different. The pulse pressure, this is in the brain. And so one of the problems with these patients is that they get not enough oxygen to the brain. So here you can see that they generally have lower flow to the brain than the control group. Another study we did was using the exact same approach to study where to place stents in vessels with pulmonary hypertension. So we imposed 20 blockages, and we wanted to impose 3 stents. And so we ran an optimization over all the possible stent locations. And you can see here with a good placement of the stent, we clearly improved the flow to this region of the lungs here and this region of the lung here. We were not able to decrease the pressure, and that's probably because it's not enough to place the stents. You also have to give some vasodilatory treatment. And finally, this is the flow in these Fontan patients to the liver. So one of the problems with these patients is they have Fontan's-associated liver disease. And so here we looked at the HLHS group and the DORV group. These are these plots here. That's the inflow to these 2 vessels. We segmented the images in the liver. The liver is interesting because it has 2 networks providing flow to the liver. And here you can see some of the predictions. And again, we saw that the HLHS patients fared much, did not have nearly as good predictions of pressure as the DORV patients. And this study is the one we are currently working on extending to understand what happens in these dogs, which are born with portosystemic shunts, which basically mean that they don't have flow going into the liver from this vessel, but it goes around. And so we have a big vet school at NC State, and the clinicians treating this disease are very interested in learning more about it using computational modeling. Now, I think, yeah, we're going to talk about this too. So another way of looking at these things is doing 0D models. So for the 1D models, we can get a lot of information about waveforms, flow prediction, wave reflection, but it's hard to do for the whole system. So in this particular study, we were interested in really looking at it from a system perspective. So we developed a circuit type model. So it consists of resistors and capacitors of the whole circuit, including the heart, the left and the right side of the heart, the atria and the ventricle, the systemic arteries and veins, the pulmonary arteries and veins. We are interested in the stroke work, which is really the integral under this pressure volume loop. And we are trying to understand the importance of this membrane that separates the right and the left heart. And so this data set is from patients that have undergone heart transplant. And one of the things we're trying to understand is, can we get more insight into how well these patients fare after the transplant? So the model is pretty simple. It's basically conservation of volume, which is that the change in volume is the flow in minus the flow out. It's Ohm's law, which says that the flow over a resistor is the pressure in minus the pressure out. We have the heart, which has valves, and then to make the whole system pump, we have a pressure related to elastance, to the change in volume. And then finally, you impose functions that make the heart contract. We have a function for the ventricle and a function for the atria. And one of the challenges here is to get the atria and the ventricle to happen, to beat at the right time. And then we use this kind of an activation function times the elastance times the volume minus the unstressed volume. It has an active component and a passive component. And so with this type of model, we used because the model is an ODE, so it is much easier to run. And I can see something funky happened to my slides, but bear with me. So here we use a Bayesian approach. We use Bayesian inference to estimate the parameters. And one of the things we learned here is that if we have a good set of identifiable parameters that we estimate, we get very nice Gaussian posterior distributions for all of our parameters, which means we can sample from these and not just get a prediction, but get a prediction with uncertainty. And the dashed line here are the estimates we got from the point-wise parameter estimation. But if you notice the scale here on our x-axis, our distributions really have very narrow intervals. And there's a reason for that, which we may or may not discuss on this slide. So in this particular study, we compared some of the patients. So one of the results we saw was when we looked at patient 266 versus patient 558. So at each of these time points, they had data measured. And so then we calculated, or we estimated parameters matching the model to the data. And then we calculated the PV loops. And what we calculated then was we calculated the stroke work, which is the area under this loop. And then we noticed that when we are going after treatment for this guy, we see that this PV loop decreases in time. And so we start out with a stroke work that is very high, and then we ended up with a stroke work that's close to 1, or power that's close to 1. We started out with a very high mean pulmonary arterial pressure. We ended up with one in the normal range. And we went back and looked at the recovery from this patient, and our model predicted this guy will have successful recovery. And in fact, this guy did have successful recovery. The other guy here, we saw the exact opposite. We saw that the power increased, and we had no data that could describe the power. So we didn't have measurements of PV loops at every time because it didn't do an echo. And so here we saw the exact opposite, that the power just kept increasing, the pressure kept increasing, and this guy really developed pulmonary hypertension, and in fact, he died 48 months post-transplant. So it's just an example of some predictions we were able to make with this fairly simple model. The next questions we were asking is the left and the right heart, they interact. And sometimes that becomes really important, but we really don't know how important it is. The literature has a very complicated differential algebraic differential equations model that we thought this is way too complicated because it's hard to solve numerically. And so we were asking, can we A, generate a simpler model, and B, can we understand this ventricular-ventricular interaction? And so the way we do that is you have the right heart and the left heart. And then you have this membrane here between the right and the left heart. And so then we said, okay, there is an area that is encountered by this. Remember, we are doing this in 0D, so there's no spatial thing. So we split the ventricle into this component, and then we say the ventricle left free wall area is this guy minus that guy, and the right side is this guy plus this guy. And then we estimated parameters for this and study what happened for this patient group. Oops, why am I not advancing the slides? Okay, so one of the things that we were anticipating was that these patients with pulmonary hypertension, their right heart grows. And when the right heart grows, it has a negative effect on the left heart. So then this P-septal volume becomes negative, which is what we actually saw for all our PH patients. And the more severe the PH, the more negative this septal volume was. And so then we did some statistical analysis, and our results basically showed that if we account for the septal volume, we can nicely separate the parameters that we estimate according to the patient type that we have. So in this study, we included data from, I think it was 20 patients. They were separated in 5 different types of patients. And so we used clustering to see, and we were very surprised that we were able to differentiate or identify clusters from each of these patient groups. There were a few patients that were placed in the wrong patient group. For example, this guy was diagnosed with pulmonary arterial hypertension, but he ended up acting like the patients that had blood clots. And it's very likely that this guy has pulmonary arterial hypertension and also had blood clots, because if he didn't have a scan, they may not have identified the blood clots. And the same thing here, this guy so this is whether they are having a heart that's functioning well or not. So this guy is acting kind of like the patients that have a heart that's not pumping normally, even though he was diagnosed as having a heart that does pump normally. Maybe he was on the borderline of this group. But all the other patients ended up in the groups that they were supposed to be at. If we did the same analysis on the data alone, it was not possible to group the patients if we did not include the ventricular-ventricular interaction. Again, we were not able to group the patients. So in the final 5-10 minutes, I'm going to talk about this different project. So in addition to, I know that you were mentioning that I did my postdoc, I did a stuff with the neural system, and really that also has an effect, even though it's not in the brain, it still has an effect on the blood pressure and blood flow. So you probably all know what happens when you start running, your heart rate goes up, or if you go standing up, sometimes you feel lightheaded or dizzy because the blood flow really needs to interact with the neural system to control heart rate and blood pressure. So I know this is a really complicated slide, but let's try and walk you through what's happening here. So when you go from lying down to standing up, you can see from this guy that we are shifting volume from the upper body to the lower body. As a result, the blood pressure in your aortic arch will decrease. There are neurons sitting on the aortic arch and in the carotid sinuses. They are stretch-sensitive neurons, and so they sense changes in blood pressure. It's part of the autonomic nervous system. They send signals to the nucleus solitary tract, which is in the bottom of your brain, where it's integrated and then transmitted via the sympathetic system and the parasympathetic system. So the sympathetic system sends acetylcholine via the vagal nerve, which is a very fast response and has an impact of heart rate. And so a change in blood pressure will impact a change in heart rate within like 1 heartbeat. The sympathetic nervous system is mediated by noradrenaline. It goes via the sympathetic pathways. They go through the spinal cord. They go to all your small vessels in your muscles. They also go to your heart, and they can also make heart rate go up. It's often called the fight and flight response. And so a lot of functions with autonomic dysfunction of some sort, POTS, and recently we have spent a lot of time studying patients with post-COVID because it's not really known why so many of them exhibit chronic fatigue syndrome. And so what we're doing is developing a model to try and study that with a specific test, and we're going to talk about that test. So now we're going to play this video, and it might talk.
This is a video of the Valsalva maneuver, and the subject should be
I don't know if you can hear it.
The time and the pressure, and hit 40 millimeters of mercury in 15 seconds. So the subject starts with an inhalation, and we start the clock.
And point out here, I'm breathing to 40 millimeters of mercury. It's actually really hard to do. Okay, so this test, the clinicians really like because you can do it at bedside. You don't have to get out of bed. My husband recently had knee replacement surgery, and they actually did a sit-to-stand test before they let him out of the hospital. Part of the problem with that is that if you're not able to control your blood pressure, you will fall, and that's not great if you just had knee or hip replacement surgery. So Anna Maria, that you can see here in the back, she's an anesthesiologist, and they're really interested in running this model and studying this test for everyone who goes into surgery before they have the surgery to figure out if their control system is working. It's also used for patients with chronic fatigue syndrome or some of these other autonomic dysfunctions. So it's very versatile, but it's really hard to clinically interpret the signals. So we have developed a new software. It's just recently published in Journal of Visualized Experiment. You can see a really long video of it, but I'm only going to talk to part of it. So part of it is that the clinicians really like to get a smart way to extract markers and features of this signal. Another part is a model that predicts the autonomic function, and that's what we're going to talk about today. So the model starts by the measurements of systolic pressure and the intrathoracic pressure. So that's the pressure they measured on this little device. It looks like this. It's superimposed with respiration. We are not going to talk about how we clean up the data to run the model because that would take another 45 minutes. But really, this pressure and the intrathoracic pressure is felt by the aorta, which is here, and the carotid, which is up here. Now, since you are holding your breath, you are putting a lot of air in your lungs, so the pressure sensed inside your thorax is different than the pressure sensed up in the aorta. We are using a first order set point differential equation to model the strain that the neurons are sensing separately in the carotid and the aorta. Then we are integrating the signals from those two locations in the brain. We are combining that with the signal sensed by the respiration and predicting the sympathetic and parasympathetic and respiratory driven parasympathetic signals. Again, this is done by a system of differential equations. Now here is an important component. It's a delay differential equations because the parasympathetic system is immediate. So for that, the delay is 0, but the sympathetic system is delayed. And so we need to take that into account. Again, a first order differential equations with a health function, which is some kind of a set point here. Everything saturates in biological models. Then we are predicting heart rate. So we have an intrinsic heart rate, which is a signal the heart was beat at if there's no input. We have the parasympathetic response, which makes heart rate go down. The respiratory response, which makes heart rate go up. And the sympathetic response, which is delayed, which also makes the heart rate go up. Again, we have a differential equation. So summarizing this model, it has 1, 2, 3, 4, 5, 6 state variables, but it has 28 parameters. And we did the same thing we did for the other model. We did sensitivity analysis. We did subset selection. We checked that these parameters were identifiable. And then we estimated these parameters, again, using a Bayesian approach. And we were able to get a model that very nicely predicts the data. And we were able to get these uncertainty bounds here on the model prediction. Now the output here, so why do we even care about predicting a signal that you can actually measure? Well, what we care about are the underlying parameters and how they change between the patient groups. And the other thing we like to do is we like to give the clinicians insight into what the sympathetic and parasympathetic signaling might do, because it's something we cannot measure. And so we are still working on finalizing this model. But before, let me actually skip ahead a little bit. We took this model. We are in the process of running simulations, estimating parameters from 800 different patients, but it takes a lot of work to do. And then we are trying to see if we can distinguish differences between the patients. And we have in this patient group of 136 of the 800 patients, 55 with a normal response, 37 with some light dysfunction, 22 with another type of dysfunction, and 22 with a new type of dysfunction that hasn't really been described well in literature. And one of the things we were very happy about is that just looking at some of the predictions we made, we were able to see differences. Another interesting little mathematical side note on this project is that this is a dynamical system. As we said before, it has 6 differential equations. So we try to see something about, okay, what are really the essential equations and what is really the impact of the delay we are seeing in the sympathetic system. So we looked at a much simpler two-compartment system, and I'm sorry I forgot to put a reference for this study. It was published, I think, a couple of years ago with my graduate student, Ben Randall. You'll see him acknowledged in the end. So basically what we did is we just looked at a reduced version of this model. We're assuming that the parasympathetic response is immediate, that we can combine the aortic and carotid strain. We don't have to include the respiratory mediated system. And by doing that, we can get a two-state system with a sympathetic response and the response on heart rate. And then we can, but it's not an autonomous system, so keep that in mind. But we could still go in and calculate what is called a characteristic equation. And when we calculated that characteristic equation, we could study the dynamics response in the delay versus the time constant associated with the sympathetic system. And we found something really interesting, so that we found that at some value of the delay, the system undergoes a Hopf bifurcation. Now, we have an unstable focus on the left side of the Hopf bifurcation, which really would mean unphysiological behavior. So we never saw any patients that had that type of behavior. We had a region where we had a stable focus, which really corresponds or translates to an overactive sympathetic response. And some of our patients, especially the POTS patients, seem to all fall in that group. And really, one of the things that characterize POTS is postural orthostatic tachycardia. These are patients that whenever they move, their nervous system overreacts. And then the normal patients found in this group here, which was a sync. And so we had another bifurcation here along this line. This was our theoretical model. This is simulations with our actual model. And so we were really nice to see that you could do this kind of dynamical systems analysis and really see different types of behaviors for these patients. Okay, so in summary, starting these differences, we really saw differences between the patient groups, and there's not currently any constructive way to actually analyze these signals. So while the clinicians are really excited about moving on with this type of software, there currently does not exist a software that can do that. And we are trying to think about how we can move this project forward, establishing more closely connections between the results that we get from this type of dynamics model and the measurements that you create in the clinic. So I didn't do any of this work alone. I really want to thank all my collaborators. So the 1D modeling stuff is done with Charles Pulse at University of Houston, Nick Hill at University of Glasgow, Dirk at University of Glasgow, he's a statistician. Tom Chilemsky is an MD at VCU who's very interested in the Valsalva maneuver. And Sujit is a faculty member in statistics that I recently started working with. Anna Maria is an MD. Alan is also one of my collaborators who's very good at sensitivity analysis. I have a bunch of students that already graduated, including Alyssa, Mitchell, Ben, and Tess, and Megan, and Michelle, and some current students that are contributing to this work. And I want to especially thank our funding from the NSF in form of 2 big training grants, one for graduate students and one for undergraduate students. And then we also got some research funding from both NSF and NIH, and most recently from the Additional Ventures Foundation. So thank you very much.
Thank you very much. Fantastic talk. Questions? So before Anmar, go ahead.
I have a couple of questions. For your first part of your talk, you talked about skeletonization and these artifacts that have been generated. Out of curiosity, how did you remove these artifacts? Maybe you said it and I missed it. How were they removed manually?
I reached over it very quickly. We actually spent a lot of time working.
So you're doing it manually?
Well, partially. some of the artifacts we are removing manually. So basically, these extra edges we are removing manually. Identifying loops we can do automatically, because you just look at the degree of this branch. If it has degree 4, it's a loop, and we can go in and find it and remove it. We just remove the branch from the largest vessel to the smallest vessels. There's no user interaction in that. Keep in mind, this network has 3,000 or 4,000 vessels, so you don't want to do it manually.
Yeah, that's why I'm a little bit surprised, because I was thinking that the branching is very complex.
It worked really, really well, and it's a very challenging problem. Another thing we often see is in junctions, you get these small cycles. Again, these small cycles are easy to detect, and we use the Discuss algorithm to remove that manually. It's described in detail in this. Another thing we have added to this, which I didn't talk about, is if you're not careful when you're segmenting, we are creating what we call blobs, which are holes in the segmentation, which confuses the skeletonization algorithm. And so we detected we generated a blob remover, which is partially automatic. So it detects the blob, it shows where they are, and you have to click on a node in the blob, and it goes in and creates just an edge where the blob was. So we haven't yet published that, but that's something we have worked a lot. John, who's one of my undergrads, have worked a lot on automating this, and Megan, which has graduated, has also worked a lot. So the software we are using for the skeletonization was developed by Pablo Hernandez, and he developed this code called SegEx. It's available on GitHub, but it wasn't made for networks, but we spent a lot of time adapting it to use, and it has really worked fantastic in any of the networks we're doing, and it works much better than VMTK, which really doesn't do well on large networks. And I'll be happy to answer more questions about it if you have that, because it was hard.
Okay. Yeah. My other question is regarding the last part of your talk. I mean, with this delay differential equation model, you mentioned towards the end that you're ending up with this oscillator regime when you cross a hop. So when you're using Bayesian inference, what guarantees do you have that you don't end up in this oscillator regime?
I know. This is my biggest question. So we're still working on the Bayesian inference part with the statistician, and I always ask them, what happens when you have a hop in your data, and does that happen in your parameter range? I think the parameter range that we are looking at, we are not seeing the hop bifurcation, but that's still an open question, and it's something I'm very interested in, because the Bayesian inference is not, there's not a lot of literature talking about what you do when you have behavior like a hop. And so currently we are solving it with the Bayesian methods without the delay. I'm not happy about doing it. We have big discussions of whether the delay is important or not. I try to see that it is, because it does generate different types of behavior, but it's work still in progress, and I'm very excited you asked that question.
Okay.
And
Thanks.
answers to come, hopefully.
Any other questions? I had a related question, actually, very naive one in my case, because I'm not in this field. But you mentioned briefly that it's very difficult to do the fitting for the 800 patients. I was wondering what the source of the difficulty is. It individualized? Where is the individualization coming in?
The difficulty comes in that you have a lot of parameter correlations. And so if you have a model, I don't know, do I have that? I took that slide up. Okay. When you have a model that have this many parameters, and the data is often messy. So I showed a really good results from a normal patient. Now, the patients that are not normal, say, for example, they don't see an increase here. If the model is expecting an increase, but the data shows a decrease, we are still working on figuring out, can we still use the exact same model? Does it work? The disease case is challenging. So I would say a lot of the challenges come with cleaning up the data before you get started. And so the majority of this work, which is what Tess worked on for a master's, was really developing the software that does all this first part that I completely skipped over. But the cleaning up the data is essential to be able to make good model predictions. Now, when you're cleaning the data, you need to make sure you're not cleaning things that are physiological responses. And we just are collaborating with VCU. They just hired a postdoc. Our postdoc is going to use our model. And so he has a physiological insight to know what to do when you are cleaning the data. And that's really, I think, the big challenges here.
So you're making your way through the data set, keeping in mind the prediction known, and essentially fine-tuning your model so that it will work well on a new data set, right?
Correct. And the other thing we have recently done that I didn't talk about either, we did a transfer learning approach for the Valsalva, the front-end study, because now we want to do this for 200 patients. We need to come up with efficient ways of doing it. And we have some collaboration with the statistician to think that approach might work. But that's still work in progress. Yeah. But there's a lot of challenging doing this on real data. I would say the biggest challenge is when you're not working on example data, but are working on real data, it's just hard because it doesn't always work the way you want to work it. The nice, complex theoretical methods, some of them work, some of them doesn't work. Like the question that came up with the hop bifurcation, that's a really question very close to my heart. How do we make sure that that the Bayesian approach works, that the statisticians love, but we don't always use it? But it's great because it gives us uncertainty bounds in a smart way.
Yeah, you also briefly mentioned that the statisticians didn't like the constraint of the volume constant volume that you placed on the model. I'm surprised. I thought it would make sense that you would have such a constraint given noisy data sort of to regularize the predictions.
Yeah, that's correct. And sometimes you do need to make some assumptions because you don't have information about everything.
Okay. Other questions? Okay. If not, then thank you very much once again. Fantastic talk, really interesting. And I might reach out to you because I have some follow-up questions.
That's perfectly fine. And if you feel free to send me an email, if you have any questions or one thing you want to discuss, I'll be happy to talk about it. I think the main thing to keep in mind is if you want to work on real problems, you have to use more types of math, and you really have to learn to talk to the people or the clinicians who have the data and what are the questions they're interested in.
So there is a question in the chat. Just to know what feature the sensitivity analysis was done on, integral of the pressure loop or something else?
It was done on the data that we had available. And so for that study, the data we'd have came in terms of electronic health records. So they were really just maximum and min values of pressures and flow. So these patients had a pressure catheter, so they had measurements of pressure at different locations. So we created a least squares cost that consisted of some of these measured values that came from the electronic health records.
Super. Thank you so much then. Thanks everybody for coming, and thank you, Mette, for the talk. Bye.
Thank you.